Hello guys, in today's lesson we are going to learn about electron transport chain. This electron transport chain present in chloroplast as well as mitochondria. We have studied in chloroplast in intermediate level. You all remember that it is non-cyclic Z scheme. Do you know what is the purpose of ETC? Let us know in the class. If we blow excess gas in a balloon, the balloon gets high pressure. The air comes out with high pressure. Like that, proton gradient increases one side. This gradient helpful in synthesizing ATP. It rotates the ATP synthase so that ADP plus inorganic phosphate get mixed up and forms ATP. I think you have understand now what is the purpose of ETC. So what is the purpose of ETC now? Increasing proton gradient in thylakoid. Also electron flow from the H2O. So did you understand what I told you? Increasing the proton gradient in thylakoid so that the pressure is helpful for creating ATP. Okay, let us start our lesson now. Introduction to electron transport. What is electron transport? Electron transport is a series of redox reactions where electrons move through carrier, occurs mainly in thylakoid membrane, mitochondrial inner membrane. So I told you ETC happen in mitochondria as well as in thylakoid. Main goals: convert light or chemical energy to electrochemical gradient. So we are forming proton gradient here, H plus ions, okay? Generate ATP via chemiosmosis. So one side protons are increasing, these protons go outside with the pressure. So chemiosmosis happens here. By this ETC, NAD get reduced by protons. So NADPH, NADH get formed. In the plant physiology, we have already studied about chloroplast ETC cycle. So here also we see that only. So let's go to second point. Location in plants. Thylakoid membrane architecture. Okay, it is happen in thylakoid membrane, embedded in protein complexes. Photosystem 2, cytochrome B6F complex, PS1, photosystem 1, ATP synthase, low proton concentration. Compartments means rooms, chamber. Stroma has low proton concentration, lumen has high proton concentration. So this is different, helpful in forming ATP. Third point, components of photosynthetic electron transport. What are the components available? Let us see. Major electron carriers, photosystem 2, that is P680. It splits two water molecules, two oxygen, 4 protons, 4 electrons. But in the diagram, we see one water molecule of O2, 2 protons, 2 electrons only. So because it shows only one water molecule, here it happens with 2 water molecules. Please remember this. Second component, that is plastoquinone. It is a mobile lipid carrier. So it moves one place to another place. Transfer electrons from PS2 to cytochrome B6F. Okay. Also carries protons into lumen. Please remember this. First to watch all this. Later we will see the diagram. Third component, cytochrome B6F complex. Its functions similar to mitochondrial complex 3. Performs Q cycle. We will see it later. Okay. It pumps protons into lumen, hydrogen ion to lumen. Fourth component, plastocyanin. It is a copper protein. So please remember it is a copper protein. Transfer electrons to PS1. Okay. Fifth component, photosystem 1. It is a reaction center. It is P700. Okay. Boosts electron energy again using light, because P680 electrons are insufficient. So we need to boost more electrons here. Sixth component, ferredoxin. It is soluble iron sulfur protein. Remember it to transfer electrons to ferredoxin NADP reductase. That is FNR. So NADP gets electrons plus protons and forms NADPH here. We studied it in previous slide. Okay, now you can see the diagram. It is non-cyclic form. Okay, you can see minus 0 and again minus. Okay, it is a redox potential, means voltage. Okay, here is negative voltage, positive voltage, negative voltage. Okay, please remember this. So P680 is in negative voltage here. Okay, it gets electrons from the water. I told you that one water molecule is shown in the diagram. So you can see that H2O forms half O2, 2 protons, but in reality it happens with 2 water molecules and forms 1 oxygen, 4 protons, 4 electrons. Okay, please remember it. Here you can see that photons exciting the P680. So P680 get excited here. Electrons are moving from it. So it loses electrons. These electrons should be covered by water here. Okay. These excited electron moves to pheophytin, plastoquinone, cytochrome B6CF complex, plastocyanin, P700 reaction center. Again it go to ferredoxin, NADP, and NADPH get formed. Okay, because P680's electrons are insufficient while moving, they lost their energy. Okay, so we need to boost the electrons in PS1 also. Let us go to next slide. In the diagram you have to remember minus points. Okay, it got mistake, so please remember it. These are minus. You have seen in previous slide. Please watch it. It is a fourth step, that is linear electron flow. Light excites in PS2, water splitting replaces the lost electrons. Electrons goes to plastoquinone, cytochrome B6F, plastocyanin, next to PS1. Again PS1 absorbs light, again releases electrons. These electrons goes to ferredoxin. NADP get reduced here and forms NADPH. So ATP forms, NADPH forms, and oxygen get released from the water. PS2 does not have cyclic form, but PS1 have cyclic form. So the electrons come back and form cyclic shape. Okay, electrons come back from the ferredoxin to cytochrome B6F. Okay, so here NADPH won't form and only ATP get generated here. So why this CF should be happen in plants? It is required to protect from the excessive light. These electrons should come back and balance ATP NADPH pool. Okay, so it protects from the excessive light. Sixth point, proton motive force and chemiosmosis. I told you in first slide that protons are accumulated in thylakoid. Okay, protons accumulate in the lumen due to water splitting at PS2. Here is some reasons why protons are accumulating in the lumen. Okay, because water is splitting, because of the Q cycle in cytochrome B6F. It is required for NADPH formation in stroma. ATP synthesis happens with ADP plus inorganic phosphate. Okay, the concept was introduced by Peter Mitchell in chemiosmotic theory. Now seventh point, we have discussed about this in previous class. Please watch it also. Regulation of electron transport. Why it should be regulated? Because balance should be happen to protect from the excessive light. Okay, so now we read it. It controls PS1 PS2 excitation balance. State transitions we have discussed in previous class. LHC to migration adjusts energy distribution. Non-photochemical quenching. Excessive light get removed by heat. Redox regulation. Ferredoxin thioreduction system. Let us study it later. Here we are not studying about mitochondrial etc. But let us see the differences. What is the electron source in chloroplast etc? Water. Okay, in mitochondria it is NADH FADH2. What is the final acceptor here? NADP plus. It accepts protons, also it consumes electrons. Okay, here it is oxygen. Do we need light here? Yes, we need light here. We do not need light here because it is respiration. Which side ATP get formed? It formed in stroma here. It formed in mitochondrial matrix here. Let us see Z scheme in little depth here. What is Z scheme? The Z scheme describes the change in redox energy electron potential. Okay, as electrons move from the water to NADP plus through two photosystem, that is PS1 and PS2. This is called Z. So it is like Z. Okay, that's why we call it as Z scheme. It is in zigzag order. Okay, here electrons are moving as zigzag order. What is the core idea of it? Electrons are boosted 2 times by the light. Okay, first boost happen in PS2, second boost happen in PS1. So without this boosting, NADPH won't be formed. Okay, second point, energy axis of Z scheme. We show in y-axis redox potential. We have seen it. Sequence of carriers shown in the x-axis. Okay, redox potential logic. If there is more negative potential, there is high energy. Okay, so here light pushing the electrons to higher energy levels here, right? Third point, oxygen evolving complex, that is OEC. Reaction happens, we already know that. 2H2O gives rise to oxygen, 4 protons, 4 electrons. So water has more electrons here, so it is having higher redox potential. Here electrons have lower energy. At the water level there is a low energy. Fourth point, first energy boost. It happens in PS2. P680 photosynthesis system get excited here. Here electrons moves to higher negative potential. First electron acceptor, that is pheophytin. Electrons moves to quinone A to quinone B. After P680 loses electrons, it becomes P680 plus. So it needs electrons now. Those electrons comes from the water. Fifth point, downhill energy flow to cytochrome B6F. You can see all of these in diagram, okay. Sixth point, we already know this. So I am directly starting from this point. Energy level becomes extremely negative here, minus 1.2 volt, okay. This is the highest energy peak in the jet scheme. So it reaches to the highest peak here. We already know finally electrons moves to NADP plus. The purpose of jet scheme is for this only. Eighth point, complete energy flow summary. Low energy electrons from the water, light rises energy at PS2, gradual energy loss, light rises energy at PS1 again, energy used to reduce NADP plus. We can see the redox potential values. You have to remember these all. Left side there is a component, right side there is its redox potential. Water or oxygen have positive 0.82 volts, okay. P680 have positive 1.1 volts. Phylloquinone have 0. Plastocyanin have 0.37 volts. P700 have 0.43 volts. Ferredoxin minus 0.3 volts. NADP plus and NADPH has 0.32 volts. So remember it. PS2 is a strong oxidant, PS1 is strong reductant. It is the last slide, please watch it. 10th point, that is thermodynamic logic behind jet scheme. Why two photosystems are required here? Because it cannot happen with single component here. Single photon excitation cannot span energy gap from H2O to NADP plus, okay. Energy requirement, we need 1.14 volts to do this action. So here it is pushing the electrons to one side to create 1.14 volts. It is selected by evolution. Okay, two light reaction, stepwise energy. That's why there is two photosystems, stepwise energy flow. Common CSIR-NET questions. Why P680 is stronger oxidant than P700? Because P680 has extremely positive redox potential. So it loses electrons efficiently and becomes reductant. Which step generates electron gradient? That is phylloquinone reduction plus cytochrome B6F Q-cycle. What is the highest energy electron location? That is after PS1 excitation. We have completed our class. If you like my videos, please subscribe my channel. I try to finish 4 to 5 units until CSIR exam.